Hello guys, today's presentation is about transmission of microbial diseases. We are going to see how the microbial pathogens gets transferred from one person to another person and causes diseases in the society. The number of slides are more, but I will try my level best to make this as short and sweet as possible. Before we see the details of transmission of diseases, let us first understand a few terms connected with it. The first one is source. A source is the location from which the pathogen is immediately transmitted to the host. That means the pathogen is residing in a place, in a location, and from that location the pathogen gets transmitted to the host, causing the disease. So that location is called a source, and this source can be either an inanimate object or an animate object. Animate in the sense it could be animals or human beings. Inanimate environments like water, soil or food can act as a source of a pathogen from where the host gets the pathogen inside his body. And there is another term called a reservoir that is also used. What is it? The reservoir is the site or natural environment in which the pathogen normally resides. Now, what is the difference between these two? Source is also a location. I am telling reservoir is also a location. What is the difference? See, I will just explain it with an example. Let us talk about hepatitis A virus. This virus causes a disease that is hepatitis or type A hepatitis to be specific. Now this hepatitis A virus normally exists in a human body. Sometimes it causes disease, sometimes without causing a disease, it can normally reside in a human body. So the reservoir for hepatitis A virus is humans. Whereas when it causes disease, what usually happens is from the reservoir, that is from the human body, the virus gets shed in the fecal material, and this fecal material contaminates a water source, and when that contaminated water is consumed by another human being, he can get the disease. So now the contaminated water is the source for hepatitis A virus. So can you make out the difference now? A source is a location from which the pathogen is immediately transmitted to the host, causing the disease. Whereas a reservoir is the site in which the pathogen normally resides. Sometimes reservoirs can also become a source, can also function as a source. That is, from a human being, another human being gets the hepatitis disease. So here the human being who is the reservoir also acts as a source. And again, reservoirs also can be animate or inanimate objects. For example, we talk about tetanus disease, which is caused by Clostridium tetani. Clostridium tetani usually will be present as spores in the soil. So that is the reservoir of Clostridium tetani. Soil is the reservoir of Clostridium tetani. So reservoirs can be animate or inanimate objects. How the disease gets transmitted? There are various vehicles by means of which the disease can get transmitted in the society. By contact, either direct contact or indirect contact with a source, by inhalation, by ingestion of pathogens, by inoculation, by certain insect vectors, and also by congenital route. What are all this? We are going to see in detail. So the first one among the route of transmission is a direct contact. Under direct contact, we have two types again, horizontal transmission and vertical transmission. We will see vertical transmission later. First, let us understand what is horizontal transmission. See, in direct contact, if the disease gets transmitted from one person to another person directly, that is called direct contact disease or person to person transmission or horizontal transmission. And this horizontal transmission can take place again by three ways. The first way is where the human being comes into close contact with someone who is infected or who has the disease. Close contact in the sense it could be just a handshake or kissing an infected person can spread the bacterial cells, viruses or other pathogens to an uninfected person. Or sometimes because of some animal contact also you might get the disease. For example, rabies, leptospirosis and toxoplasmosis. Here the direct contact with an animal is necessary. An animal bite or scratch by an infected animal can spread the disease to an uninfected person. The next is through sexual intercourse. See, the exchange of body fluids such as through sexual contact is another example of direct contact transmission of diseases like gonorrhea and AIDS. And sometimes direct transmission of disease from person to person can involve violent expulsion of respiratory droplets through sneezing, coughing or simply talking. So here the respiratory droplets, if it is inhaled by a person who is standing very close to an infected person, then sometimes that way also the disease can get transmitted. So examples are many here. Again, what we are seeing right now, the pandemic is also an example for this. But a very common example what we see is common cold and influenza. While talking about person-to-person contact, there is another term we will always come across, and that is carriers. What is a carrier? A carrier is a person who has recovered from the disease but continues to shed the disease agent for quite a period of time. For example, a person who has recovered from typhoid fever becomes a carrier for many weeks after the symptoms of the disease have left. The feces of this individual may spread the disease to others through contaminated water or food. Again, there are four types of carriers we come across usually in the society. The first one is an active carrier. Who is an active carrier? He is an individual who has an overt clinical case of the disease. That means he is suffering from the disease, at the same time he is shedding the pathogen in the environment and transfers it to others. So such a person who suffers at the same time he suffers from the disease and also spreads the organism, such a person is called an active carrier. Whereas a convalescent carrier is an individual who has recovered from the infectious disease but continues to harbor large numbers of pathogen and sheds it. That means a person who has suffered a disease, recovered completely from it, but still he is shedding the organism in the environment through the fecal material or through droplets or through some other means. So such a person is called a convalescent carrier. The third one is a healthy carrier. This type of carrier is very dangerous. He is an individual who harbors the pathogen but is not ill. That is, though he has the organism in his body, he is not suffering from the disease, but the organism is shed by him for quite some period of time and gets transmitted in the environment. So such a person is called a healthy carrier. You will never know is the person is having the organism and also shedding the organism in the environments, still he will be doing it. And the last one is an incubatory carrier. Is an individual who is incubating the pathogen in large numbers but is not yet ill. That is, a person has got the pathogen in his body, the pathogen is multiplying in his body, he has not shown any symptoms yet. That means he will be showing the symptoms later. But during this period, which is called the incubation period, he will start shedding the organism in the environment. So such a person is called an incubatory carrier. Again, based on how long he will be shedding the organism, transmitting the organism in the environment, we have two terms again: transient carrier and chronic carrier. Transient carrier is a person who sheds the organism for a few weeks or 6 months, for a short period of time. When he is going to shed the organism in the environment, then we call him as a transient carrier. Whereas a chronic carrier is a person who could shed the organism for years together. For 5 years to 10 years, if a person keeps on shedding an organism, such a person is going to be called a chronic carrier. Indirect contact refers to the transmission of the pathogen from the source to the host through an inanimate object. And we call these inanimate objects which act as a vehicle of transmission of disease as fomites. Fomites can be anything, any inanimate object. For example, if the disease gets transmitted in children through fomites, usually the fomites will be the materials that are shared by the children, like pencils or toys. This may be contaminated by the pathogen from one child and gets transmitted to another. But if you see it in the adults, this sharing ability is lesser. So how inanimate objects acts as a vehicle, acts as a source, mainly with the objects like doorknobs or telephones, push buttons or faucet handles, which are usually handled by many people at a time. Even shared bed linen, bed materials, bed sheet or bedspreads or pillow covers, through this, or by sharing handkerchiefs or sometimes toilet seats, toys, eating utensils, clothing. When you share clothing, personal clothing, or syringes also, sometimes drug addicts, they tend to share the syringes. So through all these inanimate objects, when the disease gets transmitted, that is indirect contact. The next method of transmission is vehicle transmission, where the disease gets transmitted either through contaminated food or contaminated water or through air, that is through aerosols. So there are certain examples I have given here. Let us see one by one a little more in detail. The first vehicle transmission that we are going to see is airborne transmission. Here the pathogen is suspended in the air through sneezing, coughing or talking by an infected person, and the pathogen travels to some distance, over a meter or more, from the source to the host. And when such a pathogen is inhaled by a healthy person, he might get the disease. And the pathogen that we are talking about here can be contained in the air within a droplet nuclei or within dust. So there are two different vehicles that we are talking here again. The first one is droplet nuclei, the second one is aerosols. What is the difference between the two? Let us see that. Droplet nuclei can be small particles about 1 to 4 micrometer in diameter. It results from the evaporation of larger droplets. See, when the person sneezes or coughs, the droplets will come out and get suspended in the air. These droplets will be usually larger in size, that is, they will be 10 micrometer or more in diameter. Quickly, these droplets will get dried up in the air, become smaller in size, and they will become 1 to 4 micrometer in diameter. So when they become smaller in size, we call them as droplet nuclei. So droplets usually will get settled down on an object quickly when compared to droplet nuclei. Droplet nuclei can remain airborne, can remain suspended in the air for hours or days together, and they can also travel long distances. So what is dangerous here, droplets or droplet nuclei? Obviously droplet nuclei, isn't it? Because they are smaller in size. Since they are smaller in size, they will remain suspended in the air for many days together, and they can also travel longer distances when compared to droplets. For example, diseases like tuberculosis and influenza usually will get transmitted in this way by means of droplet nuclei. They will get mixed with this droplet nuclei, and then they will travel long distances, and then they will infect another healthy individual. Now, what are aerosols? These are suspensions of fine dust in the air that contain the live pathogens. There is no, or usually there will not be moisture. They are usually fine dust particles on which live pathogens will be adhering, and they will travel longer distances again. And the pathogen that is usually seen in such aerosols is organism that causes psittacosis, which gets mixed up in the aerosols from infected birds. Infected parrots are usually observed to give rise to aerosols in the air, and that causes a disease called psittacosis. This is just a photograph showing you how the droplets come out when a person sneezes. Each droplet is about 10 micrometer in diameter, as I said before, and initially they move about 100 meter per second. By that force with which the person sneezes, they can move about 100 meter per second or more than 200 miles per hour. So the next vehicle that we should be knowing is contaminated food and water. Consumption or ingestion of food or water contaminated by pathogens can cause disease. We know this already. So foods can be contaminated during cooking or during handling. That means during serving. Or they may be contaminated, or they may become dangerous when made from diseased animals. For example, the chickens might be suffering from salmonella infection, and when such a chicken is used for making chicken biryani, and if that chicken biryani is not properly processed or cooked, then that itself become a source of salmonella, resulting in salmonellosis in human beings. Okay, now the next is contaminated water. Water can be contaminated by the fecal material or urine from a person who is carrying the disease, who is carrying the pathogen. So there are many diseases that you will come across in your subjects. For example, Salmonella typhi, which causes typhoid, cholera vibrio or vibrio cholerae, which causes cholera. Viruses like hepatitis A, polio virus, which causes viral diseases with the same name, and also protozoan diseases like giardiasis caused by protozoans like Giardia lamblia. All these are diseases caused by the consumption of contaminated water. And as I said, water will be contaminated either by fecal material or urine from an infected person. While talking about water transmission, we will always come across another term called fecal oral route. What is this? This is another way by which transmission occurs. Here, a fecal carrier, that means a person who is shedding the organism in his fecal material. A fecal carrier contaminates food during handling, and a normal person ingests such a contaminated food. So fecal oral route is where the fecal material from a infected person gets mixed up either in food or in water or sometimes in any inanimate object. And if such a contaminated food or water or an inanimate object is handled by another person and by means of which he gets the disease, that route is called fecal oral route. That is, the organism gets shedded through fecal material and it enters the body through oral route. Many examples are seen under this. For, as I said before, hepatitis A virus through contaminated food, or amoebic dysentery, shigellosis, and typhoid fever are often transmitted this way. Sometimes pathogens can get directly inoculated into the tissues of the host by either of these three ways. That is by animal bites. For example, rabies virus enters the body, human body, through dog bites. The second one is by insect bites. This we will be seeing separately in the coming slides. This is also called vector-borne infections. And the last one is by contaminated surgical equipments. For example, through needles or through catheters or through scopes. If the organism enters into the tissues of the host, then that also is called inoculation. And this we have already seen. The iatrogenic infections we have already seen in the previous presentation. Now let us see a little more in detail about vector-borne diseases. So what are vectors? Vectors are living organisms. They could be either insects or vertebrates like dogs, cats, bats, etc. These vectors, they carry the pathogen from one host to another host, thus causing disease. Now, when the vector is specifically insect like mosquitoes or ticks or fleas or lice, then that is called arthropod-borne infections. Now, the vectors can be either a mechanical vector or a biological vector. Mechanical vector are those arthropods which passively transport the pathogen on their legs and other body parts and transmit it to a person. See what is meant by that? Here the vector, that is the insect, is going to transfer the pathogen from a source to a host. But the pathogen is not going to grow in that vector. It will be just carried by the vector. For example, houseflies. See, houseflies, what they usually do, they will go and sit on the fecal material. They will also go and sit on the food that you eat if you keep it open, right? So when they sit on the fecal material, they accidentally carry a pathogen which can be present in the fecal material on their legs. And they are not going to wash their legs. They are just going to carry it. And when they happen to see a food, an open food, they will come and sit on that food. And unintentionally, they are going to drop the pathogen on the food. And when such a food is consumed by a healthy person, he might get the disease. Usually, Shigella gets transferred in this way. It is shed in the fecal material through flies. It enters the food, and from when such a food is consumed, the person will get the disease, that is shigellosis. Whereas biological vectors are insects where the pathogen must multiply inside the insect before it can infect another host. Here, the vector is not only going to carry the pathogen, it is also going to act like a host, an intermediate host, where the pathogen is going to multiply in number and then it gets transmitted to a person. For example, malarial parasite. This malarial parasite, when it enters the mosquitoes, inside the mosquito's body, it will undergo a specific cycle. It will increase in number. And then when that infected insect bites a person, through its saliva, the malarial parasite enters the human body. So here the malarial parasite will grow both in the mosquito and also in the human body. In such a case, the the mosquito is called a biological vector. We saw how the insects can act as the vehicle for disease transmission, isn't it? Now we will also see some animals that can transmit the infection. Animal diseases that are transmitted to human beings are termed zoonosis. Humans will contact the pathogen by several mechanisms from an infected animal. How? They can come into direct contact with the diseased animal flesh. By eating a diseased animal flesh, they will come into direct contact, or they might just drink the contaminated cow's milk. The cow will be suffering from the disease, and it will shed the pathogen in its milk, and when such a contaminated milk is consumed by human beings, they might get the disease. Or humans might just inhale the dust particles contaminated by the animal fecal material, that is animal excreta or its products, or eating insufficiently cooked infected flesh. So by all these ways, humans will get the pathogen from the animals. And usually what has been observed is domestic animals are the most common source of zoonosis because they live in greater proximity to humans than the wild animals. Usually humans will get the disease from the domestic animals rather than the wild animals because they don't come into contact with the wild animals. They usually come into close contact with the domestic animals. It could be cattle like cow, or it could be dogs, or it could be cats, or sometimes even squirrels. So from these domestic animals, pet animals, when the disease is transmitted to human beings, we call it as zoonotic infections or zoonosis. The last type of transmission I would like to talk about is vertical transmission. As I said before, this also comes under direct contact transmission. And what is vertical transmission? When the infection gets transmitted from the mother to the fetus, that is called vertical transmission, which is also commonly called congenital infections. And how this happens? See, there are certain pathogens which are able to cross the placental barrier and infect the fetus in the uterus. We know that uterus is a highly protective bag, and usually no pathogen can cross this uterus and infect the fetus. But there are exceptions. There are certain viruses which can cross the placenta and can infect the growing fetus. So when such thing happens, it may result in abortion or miscarriage or stillbirth or congenital malformations. That means proper growth of the fetus might not happen. Especially if you see the rubella infection, rubella virus infection that can happen in the first trimester of pregnancy. Andre first 3 months alli enadru rubella infection ಆದರೆ then that might result in congenital malformations. That means ಒಂದು brain growth ಸರಿಯಾಗಿರಲ್ಲ, ಇಲ್ಲ fingers ಸರಿಯಾಗಿ ಫಾರ್ಮ್ ಆಗಲ್ಲ, ಇಲ್ಲ ಇನ್ನೇ ಲೆಗ್ಸ್ ಸರಿಯಾಗಿ ಫಾರ್ಮ್ ಆಗಲ್ಲ. ಈ ತರ ಏನೋ ಒಂದು ಡಿಫೆಕ್ಟ್ ಇರುತ್ತೆ ಮಗುವಿನಲ್ಲಿ. The child may come out, but it might have some kind of malformations. So such things will happen in congenital infection. So very common congenital infections are congenital syphilis, rubellosis, AIDS, etc. So whatever I said till now has been covered in this slide, a summary slide. The microbial transmission can occur by direct contact or by indirect contact. Indirect contact, again we have two types of transmission. One is horizontal transmission, the other one is vertical transmission. Horizontal transmission can happen either from one human to another human, or from an animal to human through directly through talking or hugging or kissing, or through sexual intercourse, or by inhaling infected air, that is respiratory droplets. So examples are given here. Whereas vertical transmission in the sense when the infection gets transferred from mother to the fetus. So it can happen from the mother to the fetus, or it can happen from the mother to the newborn, just born baby. So the examples are given here. Whereas through indirect contact, microbial transmission can happen from inanimate objects called fomites, or through contaminated food, contaminated water, or through contaminated air. And indirect contact also can happen through different vectors, especially through insects. So these insects, which are called vectors, can either become a mechanical vector or a biological vector, and the infections that are transmitted by them has been mentioned here. Okay, that is it for today. Before signing off, let me ask you the question. We have been hearing about coronavirus infection for a year or more. Tell me how the coronavirus gets transmitted. Is it through droplet nuclei or aerosols? Is it through direct or indirect contact? Is it through fomites or through infected animals?